What I want to do in my 2 lectures today is bring you up to date with, to some extent, some of the history of mouse phenotyping and what is happening in mouse phenotyping in the present day. And of course, as you've already heard, the mouse is a fantastic tool for generating new mutants, putting perturbations into genes, changing genes, but of course then the critical issue is what happens from those changes, what happens to the mouse, what are the consequences for the phenotype of the mouse. And that relationship between mutant and phenotype is critical for determining what the function of genes are. So if you think about the challenges for the study of disease in the 21st century, there are really 4 of those. We do need to characterize the function of every gene in the mammalian genome. That's clearly important for understanding both genetics, biology, and disease. We need to generate mutations in every gene in the mouse genome, and I'll say a little bit about that, and you've already heard something about that. But critically, we need to characterize the phenotype of all of those mutant mice that we're generating. And there are enormous challenges to doing that phenotyping, and to some extent today I want to give you some idea of the scale of the challenge and what's required to be successful. So I think the first important point to note is that we only have mutations for around about 30 to 40% of the genes in the mouse genome. There are an enormous number of genes which have never been touched by mouse genetics and never been looked at. So we need to make more mutations. And even for those knockouts that have been studied, the data is very patchy and incomplete. If you look at the legacy data in mice, it's very dependent upon the interests of the investigator who made that mouse and mutation and actually then studied its phenotypes. So we need to move from thinking about looking at individual genes for individual areas of interest to broad-based phenotyping. This is the critical thing, but of all of the knockouts, all of the genes in the genome, this should provide a wealth of new functional information about how the genome works and what genes do. And importantly, it's unbiased. We'll be looking at all genes and trying to discover all of the phenotypes that are associated with that gene. And that's going to generate an enormous number of new hypotheses and ideas about the relationship between genes and biology, genes and disease. So, characterizing the phenotype of every mutant mice is one of the key challenges for the study of disease in the 21st century. So let me elaborate what those challenges are and how we might go about addressing them. And you can think of the phenotype information of a whole genome, a whole mammalian genome, as a 3-dimensional matrix. And this is how we look at phenotypes in mouse biology now. So along one axis, you can think of all of the mutants, a mutant for every gene in the genome, and you could perhaps think of those mutants on different genetic backgrounds and in different genetic contexts as well. Along this axis, you can think of a whole variety of tests that you might carry out on the mice, maybe hundreds of tests, hundreds of parameters, disease, biological parameters that you might investigate. And importantly, I'll come on to later, on another axis, you have the environment in which the mouse is living. What diet is the mouse being fed? What is the cage environment like? And so on and so forth. So what we want to do in terms of phenotyping in the 21st century is populate this matrix and completely populate it with all of the information that will really provide us with proper information about how body systems work. So we want to put in here, into this matrix, perhaps millions of data points. This is an enormous amount of data, but ultimately, when we've populated this matrix completely, we will really have an idea not just of how one gene affects one organ, one gene affects one function. We'll have a systems view, a complete, holistic view of how the mouse works from its genome to disease to phenotype. So this is, if you like, I think a critical model of what our long-term aim is for mouse genetics in the 21st century. So let me just revisit that with a couple of points. So we want to characterize disease models not just by organ. We want to do it through a multi-systems approach. And multi-systems phenotyping means that we've got to undertake broad-based phenotype screens, and particularly broad-based phenotype screens of disease relevance, of medical relevance, because we want to relate genes not just to biology, but to disease phenotypes. And without broad-based screens, we won't understand systems. We won't really understand how systems operate together within the body, how organs interplay with each other, and what is the outcome of genes and disease. So to do that, we need to identify efficient primary and multi-system first-pass indicators of interesting disease models. Because we have thousands of mutants, we have potentially thousands of phenotypes to look at. We've got to think of ways to phenotype a mouse that is both efficient and high throughput. This is what's called primary phenotyping. We won't be able to look at every aspect of every detailed aspect of the phenotype of a mouse, but we need efficient, high throughput phenotyping systems in order to begin to build this systems level view of how a mouse works. So to do that, there are really three key requirements. First of all, we've got to develop these robust and broad-based comprehensive phenotyping platforms, and I'll come on to that are able to deliver phenotyping information for all body systems. But importantly, when we've developed those protocols, we need to standardize them. We need to have phenotyping protocols that we can share and compare phenotype data from mouse genetic centers throughout the world. Because this isn't the project that will happen in one institution. So it's going to happen in mouse genetics institutions across the globe. So we need standardized systems whereby each institution, each mouse genetics house, each mouse genetics center is carrying out those phenotyping tests in the same way every day, day in, day out. And of course, in terms of gathering all this information, bringing it out to the wider community, disseminating it, we've got to ensure the appropriate informatics developments in describing those protocols, exchanging data, and describing the phenotypes by new ontological systems. So there's an enormous number of challenges there, and over the next two lectures, what I really want to do is say how we've been addressing these challenges and ensuring that we get to a point where we can make a mutant for every gene in the mouse genome and phenotype it comprehensively, gather all that data, put it in a central database, and then distribute that to biology, genetic centers throughout the world. Let me come back to standardization. This is absolutely critical for phenotyping. And one of the difficulties of the legacy data that you find in the mouse genetics databases at the moment, which has been produced over the last 50 years, is that most of the phenotyping that was done wasn't standardized. Not only not standardized, but the information on how that phenotyping was done is often not collected. So we don't know what the mice were fed on. We don't know what kind of cages that they were housed in. We don't know the investigator who carried out the phenotyping test. Often the individual investigator makes it different to the test outcome. So we need standardization. We need to ensure that we have rigor in how each center does these phenotyping tests. And also importantly, we need to collect all of this other information about the phenotyping testing. Who did the phenotyping test? What diet were the mice on? What was the temperature of the animal house? Et cetera, et cetera. And of course, if we have standardization, we'll have better reproducibility of test outcome. Many of you will have heard the considerable dialogue across science these days and across the journals about reproducibility of findings. And part of the reason we can't reproduce many of those phenotype findings in the legacy data of mouse genetics is that we don't know how the original experiment was done. So we need to standardize the experiments and we need to record properly how the experiments were done. But if we do that, we get better reproducibility of test outcome and, of course, better comparability of test outcome. So we can go back, we can reproduce those results, and we can build on it for the future. And ultimately, of course, if we standardize data, we can share all of the results that we get from our phenotyping, and we can put them into a unified database. So To do this, over many years, this began back in the last decade, in the early 2000s, in Europe, we really led the field in this area of building a European model which involved a whole number of major mouse programs, some of which you'll hear more about in the course of lectures today, including the EUCOM project to develop mouse mutants for most of the genes in the mouse genome, the EUMORPHIA project to develop and standardize mouse phenotyping platforms, EUMODIC to undertake a major pilot program to utilize those standardized phenotyping platforms for the analysis of mutants, and other programs to both archive and disseminate the data. But I'm going to focus, first of all, on this issue of developing and standardizing mouse phenotyping platforms, the EUMORPHIA project. So EUMORPHIA set out all the mouse genetics groups around Europe to develop and standardize a set of mouse phenotyping tests. This went into a new database called EMPRESS, the European Mouse Phenotyping Resource for Standardized Screens, which was published in Nature Genetics almost 10 years ago now. So you can see the length of time that we've been working on this and bringing it to fruition. You can still go and look at that database, well, now it's been subsumed into the IMPC effort. So all of these centers around Europe worked on developing a set of standardized mouse phenotyping platforms. And here are the kind of things that they did. I'll just give you a couple of vignettes of the importance of standardizing, ensuring that we get reliability, robustness, and reproducibility into our data. So what EUMORPHIA did was it took a whole number of different inbred strains of mice, Black 6, C3H, Bulb C, 129, and it developed a whole panel of testing modalities. In this case, we're looking at the open field test, a measure of motility and anxiety and so on. And we looked at the performance of each of these inbred strains through our standardized test. In many cases, the standardized test that we developed worked well. As you can see for open field here, the ranking, I've given percentage center time, the percentage of time that the mouse spends in the center of the arena is a measure of anxiety. And you can see that for the different inbred strains, across all of the different, there are 4 centers here across Europe that carried out this test, you can see that the ranking of the different inbred strains, some of them spend more time in the center, they're less anxious inbred strains, some tend to keep to the periphery. But you can see that the test was very reliable and reproducible in terms of the ranking of the inbred strains. So this was a good standardized test. We've now got a good SOP for how we do the open field test in mouse. So that's good. We've got that test. We all do it the same way. We all have the same protocol. So here's a test that didn't work. This is the rotarod test. This is a test, again, of mouse locomotor ability. You put the mouse on this rotating rod between these rotating plates, and really you test the mouse ability to stay on the rotating rod. So good locomotor ability is reflected in a long time of latency for the mice to fall off. So here we've got mean latency for the 4 different inbred strains, in fact measured across 5 different centers in this case. And you can see that the rankings of the inbred strains in the first pass of trying to develop a standardized test for the rotarod, they're all over the place. This is a hopeless test, just doesn't work. So we went back to the SOP, we looked at how to improve it further and to how could we get the consistency across the different inbred strains so that we can achieve this reliability, robustness, and reproducibility. So we made some modifications to the test. We modified the cover on the rotating rod, so it's now a foam cover, and we eliminated some of the aspects of the test, such as the training phase, and reduced the number of trials, and now we begin to get some consistency in terms of the ranking of the different inbred strains. So I think this is just a good vignette of how we must always be looking at our tests and standardizing them to make sure that they give the same output across multiple centers, but also recording all of the other data of how we did the test what particular aspects of the test were carried out, who carried out the test, under what conditions, how the mice were housed, and so on and so forth. So over the years, both in Europe and now worldwide, we've continued to look at the procedures by which we phenotype mice across all body systems, continuing to improve them, standardize them, make them more robust, and collect all of the metadata associated with that test to ensure that we get good phenotyping outcomes. And ultimately, to begin to fill in a standardized way, this huge matrix of data points. So that's the first challenge, and we've really begun to address and make progress in that. The second challenge, of course, is to come on and actually apply those tests in a meaningful way to many of the mutants that we have across the mouse genome. And to do this, following Eumorphia, EUMODIC, the European Mouse Disease Clinic, again a collaboration of mouse genetic centers around Europe, undertook a major pilot program to utilize standardized phenotyping platforms for the analysis of the mouse mutants that were being made by the EUCOM project in Europe, of which I think you'll hear more about later. So just briefly, though, EUCOM, as part of the International Knockout Mouse Consortium, have been producing mutated embryonic stem cell lines, which are turned into to mutant mice. And over the years, they've made knockouts for more than 15,000 genes in the mouse genome. And basically, the International Knockout Mouse Consortium, I don't have time to go into this in detail, and I think you'll hear more about it later, have been making this so-called knockout-first conditional ready allele called TM1A. And this is the allele that was phenotyped in the EUMODIC project. So in fact, it's an allele with a splice acceptor into a LAC-Z reporter molecule. There's a neocassette for selecting the mutation events in the embryonic stem cells. So this is the mutant allele. There are many genes where we have embryonic stem cells for this particular mutant allele, and this is the mutant allele TM1A that was used in the EUMODIC program. So to briefly summarize the EUMODIC program, as I said, it's to undertake a major pilot program, utilizing these standardized phenotyping tests, analyze around 500 mutants from the EUCOM resource. There were 4 major mouse centers involved with this, and the idea was to assay the utility and efficiency of a broad-based primary phenotyping pipeline. So we're looking at the logistics of mouse production and phenotyping, the utility of the assays, the sensitivity of the assays, but ultimately to capture and disseminate and build a set of rich phenotypic information for those 500 genes or so. So beginning to fill in that big matrix, that 3-dimensional matrix of phenotype information that we need to fill in to study disease systems. So here's the workflow of the EUMODIC program. We're taking mutants from, or ES cells from the EUCOM program. We were making mouse mutants out of these, germline transmittants, breeding heterozygotes, and then intercrossing them to produce homozygotes. And where the homozygotes were viable, they went into this primary phenotyping pipeline called EMPRESSslim that I'll come on to at a moment. And for each mutant gene, we put 7 males and 7 females through that pipeline. If the mutants weren't viable, we went back to the heterozygotes, and the heterozygotes went through the primary phenotyping pipeline. So this is the phenotyping pipeline that we carried out. As you can see, it covers a number of different disease areas, from morphology, metabolism, cardiovascular, bone, neurosensor, behavioral, hematology, clinical chemistry, allergy, and immune tests. So a whole variety of tests. There are, in fact, 2 pipelines here. Cohorts of mice went through each pipeline, and you can see that the tests cover a number of different phenotypic areas. In fact, there are 20 phenotyping platforms in total, and we measure on all of these 20 phenotyping platforms 413 phenotype parameters. So a whole number of phenotype parameters are gathered, but importantly, and I've already alluded to this, we also capture 146 metadata parameters. of how the tests were actually done, who the investigator was, how they were housed, what the diet was, and so on and so forth. So this is the summary of the data that came out from the project. We, in fact, ended up analyzing 450 mutant lines from 320 genes, and all of the data was entered into the database Europhenome. There are 9.5 million data points, nearly 3,000 phenotype annotations from over 27,000 mice analyzed for those 320 genes. So you can see the enormous scale of the work just in doing this number of genes. So think about multiplying that, not just by 10 times, but more than that to get up to the 20,000 or so genes in the mouse genome. We analyzed this using a Bayesian multi-level model, controlling the false discovery rate. I won't go into the details of the statistics of that. But out of all the analysis, we found that 83% of the mutant lines are phenodeviant. They have at least one highly significant phenotype annotation. But importantly, 65% of the lines have multiple phenotype annotations. They demonstrate pleiotropy. They demonstrate the multiple functions that genes often have in a mammalian organism. They're not just affecting one organ, they're affecting multiple organs. They're affecting systems across the organism. Of course, we measured things like viability and fertility. It's important to recognize that a large number of the homozygous mutants that we looked at are lethal, about 33%. Some are subviable. This is very comparable to other figures that are coming out of bigger phenotyping projects now. So 83% of the lines, as I mentioned, have an annotation, and this includes annotations for viability and fertility. We can also look at the annotations according to whether the allele was homozygous when it was phenotyped or heterozygous. Sorry, this is a bit small, but this is the latest data. And if you look at this central panel here, you can see in the dark, you can see the figures for the number of annotations and the number of lines for heterozygotes, as opposed to homozygotes, which are in the light color. But the take-home point is you can see the long tail of mutants with large numbers of multiple annotations. So we really are uncovering the pleiotropy and the wider systems that are affected by these genes. For the homozygous, about 88% of lines we analyzed were annotated, and it's lower for the heterozygotes, and that's significant. Homozygotes appear to have more phenotypes than heterozygotes. Now, this is what happens if you put all of the data together in one big heat map. These are all of the genes we analyzed across the top. Of course, you can't read them all. And these are all of the tests that we carried out down this axis. And the dark colors, either red or blue, red indicates the phenotype changing in one direction, blue indicates the phenotype changing in another direction. They indicate the highly significant changes in a particular phenotype for these genes. Up the top here, you can see body weight. There are often highly significant changes in body weight in many of these mutants. And those changes in body weight are often, as you can see, highly associated with other phenotypic changes in those mice. So these are just some of the kind of things that you can begin to see across this huge span of data. But I think it's important to emphasize again that this is data for only 450 mutant lines. This is the tip of the iceberg of what's happening in the genome. So you've got to think of this scaled up many-fold to cover the whole genome. There are 9 million data points in here filling in that three-dimensional matrix that I talked about. 3,000 annotations. Many lines have more than one phenotypic hit. Many of those phenotypes are often associated with body weight phenotypes. So there's a huge amount of information in here already for less than 500 mutant lines. Just imagine what it's going to be like when we've done the whole genome. So that's the challenge. The challenge is to produce this data and then to analyze it and uncover the genetic and physiological networks that underpin how a mouse is built, and by inference, how a human is built and how it develops. Of course, one of the things you might ask is, how good is your phenotyping? You're producing all of that phenotype data. You've standardized the phenotyping test. But do you really get the same results when you look at the same mutant across different centers? And the answer is yes. So in EUMODIC, we analyzed 22 common reference lines, and we analyzed them for meta-analytical measures of heterogeneity to look at how often did we get concordant results between centers. And the answer is that we get very good concordance. There's no heterogeneity in 62% of cases, discordance in about 8% of comparisons, but inter-center consistency was observed in the majority of instances. So our standardized tests do actually work. If you take one mutant and you analyze it in one center, you're highly likely to get the same result if you analyze the same mutant in another center. Of course, mutants that have got extreme phenotypic perturbations tend to be more highly reproducible than genes with weaker effect sizes, which are less reproducibly detected. And I've just illustrated here a reference line for a gene called MYSM1, which, as you can see, has got a lot of extreme phenotypes, and that's very highly reproduced in the EUMODIC consortium. Now, in fact, around half of the genes that we analyzed in the EUMODIC program had never been looked at before. And this is true, as I'll come on in my second talk to tell you about for IMPC. You can take a whole slew of genes. You can take 500 here, 500 there, but you'll find a whole number of, I mean, this is an extraordinary thing in mouse genetics. You can take 500 genes randomly selected from the mouse genome. At least half of them will never have been looked at in science before. A mutant will never have been made. Their transcription profiles may not be known, et cetera, et cetera. So they have no prior annotation. And in fact, the hit rate for those genes with no prior annotations was still very good. 88% of those genes with no prior annotations had significant phenotypes. This is completely new knowledge for these genes. We've never known anything about their phenotype and function before. And obviously, these genes provide us with an enormous powerhouse to search for novel disease genes, genes with novel function that might be important to look at in terms of their impact upon disease and health. I just want to come back to these graphs again and point you to this panel here, because this, you can't really read this, but these are the different phenotype areas where we have hits or annotations, going all the way from neurological and behavioral hits, clinical chemistry, down to immune, where we got relatively few hits. And in the dark, these are hits for annotations which are already present in the database in MGI, where we'd already done, there had already been some analysis of those genes. And these are the novel hits here. And you can see that the distribution of annotations for novel genes is very similar to that for previously reported and annotated genes in MGI. So we're looking at these novel genes. There are no big surprises there in the sense that, of course, we're finding diverse phenotypes. Our phenotyping pipelines are working just as well on those novel genes as they are on previously annotated genes, but we're finding a whole slew of new information where we can begin to understand what's happening with those genes that have never been looked at before. And one of the ways we can look at these novel genes is that we can classify those genes that have hits in different tests that reflect on, for example, bone phenotypes, tests that indicate that we've got a diabetic or obese mouse, tests that have got indicate neurobehavioral problems in the mice. And these are just some of the novel genes classified according to different areas where they may be impacting on these disease symptoms. And you can go in and you can look at particular mutants that are maybe hit by a number of different tests. For example, LMOD1. I'm going to tell you more about this gene later on. LMOD1 is annotated in a number of the different tests that impact upon neurological and behavioral systems. It's a gene that's never been looked at before. In fact, there's still only one PubMed hit for this gene. So what does this gene do? Well, now that we've tested it, we've put it through our phenotype pipeline, we can begin to tell what is the function of that gene and what role might it play in disease. So I want to finish up in the last few minutes of my talk by returning to if we think about phenotyping, if you like, it's the third big challenge of phenotyping. Phenotypes are not only manifested by simply putting them through a pipeline of tests, of doing an open field, taking blood and doing clinical chemistry analyses and so on and so forth. They're also about challenging the mouse. Some phenotypes can only be revealed if we challenge a particular mouse. And one of the new frontiers in phenotyping that, in fact, Roger talked about yesterday was the aging screen at Harwell, of aging the mice and revealing phenotypes in mutant genes, which hitherto could not be seen simply by putting them through a phenotyping pipeline when they're 2 or 3 months of age. And he will have talked about this and shown you, I'm going to be relatively quick about this so that there's time for questions. He would have emphasized to you the importance to look at the genetic networks involved in age-related disease, and that we need a new effort to look at the novel genes and pathways, biomarkers of age-related disease, and a greater diversity of preclinical models for aging studies. He went through all this yesterday, but we now have a Harwell aging mutant screen and overview, where ENU-induced mutations go to produce G3 pedigrees, which go through a whole slew of recurrent phenotyping from birth to 18 months. And I'm running out of time, and I want to leave some time for questions. I was going to tell you a little bit about one aspect of phenotyping in the aging screen that's of particular interest to me, but I'm going to skip over that, in fact, and really just go on to say that, of course, aging is just one of these environmental insults or challenges that help us fill up this matrix and make sure that we get all of the data, all of the systems analysis that are needed to look at gene function in every context. And those challenges in phenotyping are not just aging. We can think of other challenges that might reveal phenotypes, such as diet, putting mice on a high-fat diet to reveal obesity phenotypes, microbial status, infection, looking at the consequences of infection in different mutants, physical stress, the social environment, noise, and so on. So one of the big challenges as we go forward is not simply taking all of our mutants and putting them through a simple, comprehensive, standardized phenotyping platform, but thinking, how do we bring these other challenges to bear to reveal additional phenotypes and additional information about how systems in the mammalian organism work? So these are our challenges, and of course I'll come back to them in my second lecture. The challenges for the study of disease in the 21st century. We are generating mutations for every gene in the mouse genome, but really this is perhaps our biggest challenge, characterizing the phenotype of all those mutant mice and characterizing them in a way that we capture broad-based information, not just on the mice as they're housed day in, day out, but on the mouse when they're challenged, when they're aged, so that we can properly understand disease systems and identify new models of human disease. So I'll just, the EUMODIC program was an enormous program. These are all the people who participated in it in terms of generating the data from across the 4 centers who participated: Helmholtz Munich, ICS Strasbourg, MRC Harwell, and also the Sanger Institute at Hingston. And I didn't really have time to tell you much about the aging screen at Harwell. These are the people involved, but obviously Roger talked about this yesterday. This is what I'm going to talk about next. In fact, going beyond EUMODIC, and I'm going to tell you about the International Mouse Phenotyping Consortium and the challenge of developing a comprehensive catalogue of mammalian gene function. So I'll leave it there and take questions. Thank you.
Thank you, Steve.